Welcome to the new season of MesoTV. MesoTV is a program of the Mesothelioma Applied Research Foundation and is made possible thanks to the generous support of our sponsors. To learn more about our sponsors, please watch the following message.
My name is Kelly Batley.
My name is Clay Thompson.
My name is Neil Mowney.
We are MRHFM, a national firm with 70 attorneys and 11 offices across the U.S.
At MRHFM, we exclusively help mesothelioma patients and their families. It's all we do.
This is why we are proud to sponsor the Mesothelioma Applied Research Foundation and its work to fund mesothelioma research and provide support services and education for patients and their families.
Hi, my name's Joe Bellick, and I'm the founding partner of the law offices of Bellick & Fox. We support the mesothelioma community by providing first-quality legal representation to mesothelioma patients and their families. During these troubling times, we're proud to support the Mesothelioma Applied Research Foundation so they can continue the work that they do every day on behalf of mesothelioma patients and their families.
So, Dr. Hoang, thank you so much for being willing to join us for another episode of MesoTV. It's been a pleasure getting to know you over the years. You're at the National Institute of Health, and I'd like to take this opportunity to ask you to talk a little bit about the National Institute of Health, and with a particular focus on the thoracic oncology surgical group.
Yeah, so Mary, thank you so much. I appreciate being invited to this format and forum. It's a very unique kind of modality for outreach to both scientists and patients alike. So it's truly a unique venue, and I appreciate being here. So with the NIH, as we were talking in our preamble, it's an unusual and very unique place to both practice medicine and test out the boundaries of where medicine can go via both basic and translational clinical type research. And that being said, thoracic surgery as a discipline serves a wide community here at within the National Cancer Institute. Because as you know, many tumors that are treated here are in an advanced stage setting, and that most often involves or cross paths through the thoracic cavity, whereby there's a general kind of ongoing community level need for thoracic surgery and thoracic surgeons. So I feel like from a broad sense, thoracic surgeons here contribute a lot to just the overall community and atmosphere of the National Cancer Institute. And as I mentioned to you earlier, as of 2018, we have recently reorganized into a dedicated thoracic surgery branch, solely focused on thoracic oncology, tumors and problems, in that field. And right now, with the new group, we're headed by David Trump, who's the chief of the thoracic surgery branch. I serve as one of the principal investigators within the branch. And then we have a more junior person at a clinician scientist, early stage of their career development, Dr. Seamus Carr. And so that's the current group right now that comprises the thoracic surgery service within the NCI.
Mm-hmm. So I guess, we're all very familiar with the university setting. So what is the role of the surgeon at the NIH, and how does that compare to being a surgeon at a university setting? Is it similar? Is it different, the same?
Yeah, that's a great question. From my experience being out in academia, it's entirely different the way that we practice here. We're not dealing with the medical infrastructure and trying to push patients through in terms of more of a business-based model. Here, it's strictly devoted to answering and tackling cancer-related questions and trying to solve longstanding clinical bottlenecks in terms of limitations in treatment, poor outcomes for cancer types, of which there are many in the world of thoracic oncology. And so we work closely with many of the other clinical groups. And the unique unifying feature of being at the NIH is that everybody here who's doing clinical work is doing it from the perspective of a research objective, trying to answer a question, solve a problem, improve upon a process that is related to cancer biology. And so one of the major differences in the way that we practice is that we're working as a multidisciplinary team with other non-surgeon clinicians who are also very dedicated to a particular cancer type or problem. And that working environment is entirely different than what I'm used to. It all plays out in real time in the form of structured clinical trial, of which there are many ongoing here, both within our branch and throughout the NIH as a whole.
And I guess also another difference would be is that you're not constrained by insurance, that, any test or anything that you think is reasonable and needs to be performed to investigate a patient, you're able to do, and you have almost every service available at the NIH to be able to do, the intricate testing that needs to be done.
Oh, absolutely. And that's one of the, least known kind of facts, that is intrinsic in how patients come through, the NIH on a clinical protocol, is that if anybody from the outside qualifies for a particular intervention or research protocol or has a unique tumor type that's being actively studied, everything is actually paid for, and the constraints of Medicare and insurance and all these other logistics that I think often, too often, hamper efficient care of cancer patients, we don't have to deal with on a day-to-day basis here at NIH.
Mm-hmm. Thank you. So, I guess, the whole world of research sometimes sounds scary to a patient. Are you experimenting on me? Am I going to go to the NIH and am I going to have that standard surgery that I've heard so much about? So could you talk a little bit about maybe the different approaches and also how it all sort of melds together?
Yeah, well, once we reach like a clinical protocol or, a first in human or an early phase 1 type clinical trial, there's been a lot of background research, in the lab that has, determined and identified safety and efficacy endpoints before we get to that stage. So while in early stage clinical trials that we often do here at NIH, we are by definition experimenting to identify, the utility of a new drug, the efficiency of a new methodology, towards a problem. A lot of the safety has been built in and tested in preclinical testing and benchtop research. So while there is that kind of ongoing element, all of the clinicians and scientists here are dedicated to, in real time, work with the patients to identify any potential, safety issues and be able to navigate them through that. And it's with that kind of unique focus that we're able to kind of, truly be able to answer those questions that need to be answered in really in an environment where you otherwise may not be able to do so as efficiently.
Thank you. So I guess if we looked at a standard pleurectomy decortication, which, most mesothelioma patients are familiar with that terminology. You would be using that standard approach probably with something additive, something to see if there's a synergy either between a new medication or perhaps a different technique. But they would still have that standard of care plus or minus. Am I correct?
Absolutely, yeah. And what you're referring to, that always adhering to or incorporating that standard of care into experimental trials is what I've heard we term a lot is a window of trial or a window of opportunity trial, and those are very characteristic of a lot of the trials that we try to run here, particularly in the thoracic surgery branch, because with this particular, for instance, and obviously mesothelioma is the focus. With that particular tumor, there's been very difficult treatment options. Many different types of multimodality strategies have been tried out, and again, without putting the patient at undue risk, using a window of opportunity study design is often the most efficient to be able to quickly assess the relative efficacy and relative benefit of any kind of new therapeutic agent or new strategy along the way with standard of care to give them the best chance both for short-term good outcomes and potential long-term outcome gains.
So the window of opportunity trials are usually trials that are introducing a new agent prior to the surgical procedure to see if it has an impact of the disease, as well as allowing you to do some pharmacokinetic study to see how that drug reacts, in a patient, and does it indeed, improve the outcome of a surgical procedure.
Yes, absolutely.
So thank you. So I guess this leads me into, wondering about some of what's coming out of your lab, in the lab of people that you're working with. Are there applications in the clinical setting now, or are you working on some things that you think will have an impact on the future of mesothelioma treatment?
Oh, absolutely. And one of the things that I want to take the time to introduce to you today. So in our lab, one of the big questions that I've started back at the drawing table is how to improve surgery as a therapeutic modality for pleural mesothelioma. And traditionally, the biggest bottleneck with a pleurectomy decortication or an extrapleural pneumonectomy, so surgical intervention for mesothelioma in the chest, has always been the limiting factor of not being able to complete a completely tumor removal. And so a lot of the patients within the mesothelioma world are very familiar with the idea of macroscopic complete resection, where basically the surgeon is tasked with removing all gross visible disease during surgery. And inevitably, the Achilles heel of that time-honored approach to mesothelioma surgery is that we end up leaving small bits of tumor, microscopic deposits, invisible to the naked eye behind in the patient. And then ultimately for a large group of patients that undergo surgery as a treatment intervention, there's a high recurrence incidence, and the tumor comes back, and often it comes back and it's more aggressive and very difficult to treat and results in a poor outcome. So one of the major projects in my lab has been how to improve upon that. And over the years, there's been several strategies applied to trying to improve the effects of surgery. And so in terms of surgical adjuvants, or those additional treatments that we add on to surgery to try and improve the long-term durability of a macroscopic complete resection, has also unfortunately not been highly successful. The most common that I'm sure a lot of the patients in the community are aware of is where the after a surgery, immediately after a surgery, the surgeon will instill chemotherapeutic liquid and then oftentimes heat it up to a higher temperature. And so heated intraoperative chemotherapy is one of the most well-known surgical adjuvants that have been added on to try and increase the durability of surgery. And that, unfortunately, has not really, resulted in consistent results across surgical groups over the years, and it still remains an unproven therapeutic strategy. So in our lab, recently, what we've done is make what I would contend is a small breakthrough in potentially improving the durability of surgery someday. And one of the biggest limitations of any sort of surgical adjuvant is being able to specifically treat the tumors that are left behind. And to address that problem, what we've done as a translational research step is we've developed basically a thin film biodegradable medicated spray, if you will, that can be applied to the chest, very similar in similar fashion and sequence to heated intraoperative chemotherapy. But the major difference in this in the sprayable thin film is that we are able to engineer it to specifically contain unique anti-cancer drugs, and we can use the chemical structure of these nanoparticles that are embedded in the thin film spray to actually specifically target mesothelioma tumor as opposed to normal tissue. And this concept we've been able to demonstrate in small animal studies and with very impressive results that resulted in the recent publication in Nature Nanotechnology, one of the preeminent biomedical journals in the world of nanotechnology, and it's been very, very well received. And what I hope to do someday is translate and bring this thin film biodegradable spray as a therapeutic strategy to the clinic and hopefully be able to roll it out in human testing here at the NIH someday.
So does this spray have an affinity for mesothelin, and that's why it targets mesothelioma, or is there some other target or some other way that the nanotubes are, entering into those mesothelioma cells?
Yeah, that's a great question, Mary, and only somebody as with as deep of an experience as you would be able to ask that question. So beyond mesothelin, which is a cell surface marker, both on mesothelioma and then unfortunately also on normal cells, the nature of our tumor targeting is actually entirely different. So this thin film spray actually is engineered to actually stick a little bit tighter to cancer cells as opposed to normal cells. And all cancer cells have a property that distinguish them from their normal cell and environment by on the cell surface, most cancer cells carry a net negative charge. And so the thin film spray that we engineered is positively charged. And so it's simply a strategy of relative quote unquote magnetic attraction that allows our gel, the thin film sprayable gel, to stick a little bit tighter specifically where the cancer cells are because they're highly negatively charged at the surface. And the nanoparticles that we embed in the sprayable thin film gel are oppositely charged. So they're positive charge nanoparticles. And so when they get released from the gel, they will automatically naturally attract to a negative surface, which is the cancer cell. And we've shown in detailed mechanistic experiments in the lab that this specific targeting of cancer cells, we can drop off a large amount of drug into the cancer cell and not so much into the surrounding normal environment. So that's one major layer of how we're able to achieve targeting the cancer cell. And the other thing that's unique about our strategy is that unlike other traditional approaches that rely on chemotherapeutic drugs as a treatment, our lab has specific interest in using nucleic acids, small bits of nuclear material, genetic material, as the actual cancer drug. So it's an entirely new class of drug therapy. And these short bits of nucleic acid are termed microRNA. They are part of normal cells and they're part of cancer cells. And we found that cancer cells change preferentially change their microRNA signature for growth advantage. And so in a reverse engineering kind of concept, we can identify particular microRNA that are toxic to cancer cells, but non-toxic to normal cells. And so that's a second layer of safety and specificity that we build into this therapeutic platform that we designed with this sprayable thin film gel. So the microRNA in and of themselves, if they inadvertently get into a normal cell, they're not as toxic to the normal cell, but they're highly toxic to the cancer cell. So the use of these two biologic mechanisms, if you will, combined into a single product allows us to achieve remarkable capability to specifically target the tumor as opposed to normal tissue. And in the preliminary data that we've generated in the mice, you can deliver a large amount of microRNA to normal tissue, and the mouse seems fine, doesn't really get too sick. And in a mouse that's harboring a mesothelioma tumor, and you deliver the same gel, you get significant amount of tumor shrinkage, and that translates to improved survival of the mice group that get treated with our product.
Thank you. So a question for you then is, you've moved this into the mouse model. So what are the next steps? How do these trials, how do you build on this to eventually have a clinical trial that patients will be able to enroll in? What are the steps?
Yeah, well, so the next big steps is probably we will need to reproduce these results in a larger or intermediate animal model. And then based on some of that data, then that would be kind of the next step after that would be, approaching the FDA to show them our data, the safety and toxicity profile of the product, and then to get some sort of decision in terms of, do they think it would be safe to then start in-human trial testing. Some of the other nuts and bolts would be, we have to figure out how to manufacture both the gel itself, and then also manufacture large scale quantities of the microRNA, which are actually the drug being used in our particular strategy. These second two kind of requirements are more of a chemistry manufacturing kind of issue. And it's really establishing the safety and toxicity profile in probably an intermediate animal model that will help convince the FDA that this will be safe to test in humans. All of that being said, there is precedent for using microRNA as an anti-cancer drug in humans. There's a group out of Australia that has demonstrated the first in-human kind of proof of concept of this already, where they use a systemic delivery approach via the bloodstream to deliver microRNA in order to try and treat mesothelioma. And they demonstrated great safety profile in that initial study. And as far as I'm aware of, in Australia, they're trying to move that to, a phase 2 level setting for human testing. So we're very optimistic that, some of the technical hurdles to verify our particular product and treatment strategy, some of those hurdles have already been cleared by others, and that we would be optimistic that we would be not facing a long time to reach human testing.
Thank you. So, this all sounds exciting, and certainly we'll be following along with this research. So, for the current patient, patient who find themselves newly diagnosed with mesothelioma, has been told perhaps they're a surgical candidate, what is your approach to that patient today with the tools you currently have?
Yeah, so right now, in addition, so what I just talked about, hopefully not too far off in the future, but currently within the thoracic surgery branch, we have several active clinical trials for patients that may be eligible for surgery for mesothelioma, and we also have some trials that are for the very advanced stage, not eligible for surgery trial as well. In particular, we're trying out some epigenetic drugs that may offer added benefit to current chemotherapy standards in the treatment. And those are being run by Dr. Schrump as the principal investigator right now. We're also looking into the subgroup of folks with the BAP1 genetic mutation that run in families. And right now we're in an early kind of discovery phase of collecting patients that fit that profile to derive gene signatures that may help inform certain drugs may be more effective in terms of folks with a specific BAP1 mutation versus those that are not. So even though today we're talking about the future, there are current trials that are actively ongoing right now in recruiting patients.
Thank you. So, people travel, from, many different countries as well as the United States. So when they come to the NIH, what type of support systems are in place in terms of housing, costs, travel?
Oh, well, and now I wish I had the rest of our thoracic nursing team here to really kind of drive that point home. They're much more eloquent at expressing that. But the lodging, the travel, there's additional psychiatric, psychological, and social family support that all are part of the large NIH infrastructure here. So for those traveling as far as a neighboring state, those that are traveling cross country or across international borders, all of these services are integrated and currently available to everybody at NIH.
Thank you. So Dr. Hoang, this has been fascinating. Your work certainly is, I hope will translate to improved survivals and, better patient outcomes. And, we appreciate, any scientist, clinician who is, really dedicating their career to this disease. So thank you for staying with this disease and really pushing the envelope forward. It's much appreciated by this community. Is there anything before we conclude that we didn't discuss that you want to bring up, or?
No, and I will make one plug for you, Mary. This foundation, I'll remind you, a long time ago, we first met when I was the recipient of one of the Research Foundation Awards that funded a lot of the original groundwork for my interest in microRNA as an anti-cancer therapeutic. And so I just want to let you know, without the foundation's early support, I wouldn't be sitting here today talking about, this potentially new product that we're going to be able to test in humans in a few short years.
Thank you. Thank you very much. I appreciate you, bringing that up. So Dr. Hoang, have a, good afternoon. Certainly I look forward to continuing to speak with you, and the foundation will follow your work, and we wish you great success. Thank you so much.
Absolutely. Thank you very much.